Good afternoon, everyone, and welcome to Research Round. I'm so very pleased to announce that the trainee committee invited speaker for this year, Dr. Ivor Benjamin, has been able to join us, and he's going to present Cardiovascular Diseases in the Omics Era, a physician-scientist's journey and perspectives on health equity. So Dr. Benjamin received his MD from Johns Hopkins University School of Medicine and went on to perform internship and residency in internal medicine at Yale University School of Medicine. He also received fellowship training in clinical cardiology, molecular cardiology, and molecular biology at the University of Chicago, specifically the Michael Reese Hospital, Duke University Medical Center, and the University of Texas Southwestern Medical Center in Dallas. Since 2013, he has served as director of the Cardiovascular Center at the Medical College of Wisconsin and is appointed professor of medicine at the Medical College of Wisconsin and Froedtert Hospital. Dr. Benjamin brings more than 25 years of experience leading cardiovascular clinical and research programs and is a recipient of numerous medical honors, including the Pioneer Award from the National Institutes of Health and the Dr. Daniel D. Savage Memorial Service Award of the Association of Black Cardiologists. Dr. Benjamin's leadership of the Cardiovascular Center at the Medical College of Wisconsin is evident as he is building a transformative platform for centralizing clinical and translational research along the T1 to T4 continuum with support from the Advancing a Healthier Wisconsin Research and Educational Endowment Program. Additionally, he has a very impressive 30-year volunteer history with the American Heart Association, where he served as president in 2008 to 2009, and then the immediate past president from 2019 to 2020. As a highly regarded physician scientist of molecular cardiovascular remodeling, he is an AHA established investigator, a founding member of the Journal of the American Heart Association, and currently serves on the editorial boards of both Circulation and Circulation Research. As a chair of the American Heart Association Research Committee in 2015, he expanded opportunities in research for women, minorities, and undergraduates. Dr. Benjamin is a member of its board of directors, corporate operations coordinating committee, executive committee, international committee, and the scientific and advisory coordinating committee. He is also a longtime member of the Association's Council on Basic Cardiovascular Sciences and Council on Clinical Cardiology. Given all of these accolades and achievements, you can see why the UOHI Trainee Committee selected Dr. Benjamin, and we were overjoyed when he responded with such enthusiasm to accept this invitation. And we are so happy that despite the cancellation last year, due to the onset of the COVID-19 pandemic, we were able to schedule this virtual visit. So please join me in welcoming Dr. Ivor Benjamin, whose title is Cardiovascular Diseases in the Omics Era, a physician-scientist journey and perspectives on health equity. And just one reminder to please submit your questions using the Q&A feature. They will be answered at the end of the presentation. And if you're experiencing any technical difficulties, please reach out via the chat. And with that, I will turn it over to Dr. Benjamin.
Well, good afternoon. Dr. Mulvihill, that was quite an amazing and generous introduction. Thank you so very much. I would also like to express my thanks and appreciation to many esteemed friends, including, of course, Dr. Peter Liu. But most importantly, I am being invited to speak to you on behalf of the trainees. And that's a single honor. And I thank you so very much for your invitation. And the only regret that I have is that you may have to re-invite me again, because I will come in person if you do extend that invitation. But that being said, thank you so very much. So what I will try to do in today's, of course, I have no disclosures. What I will try to do in today's presentation is to provide a couple of perspectives through the lens of a trainee, where I was perhaps very similar to where you were. What kinds of decisions and opportunities were available, and how they have helped to spark and foster my career development, provide you some insights around my research interests, but just as importantly, I happen to be at a time in my professional career where I'm absolutely convinced that our support of science, technology, engineering, and medicine, coupled with, of course, mentorship, are so critically essential for inclusive excellence. And I hope that, of course, I will be able to convince you that there is a space for each and every one of our trainees to have that kind of flourishing, not only scientific career, but professional career that I think will be an antidote for burnout, as well as, of course, having a very healthy perspective in your own contributions. So as Dr. Mulvihill pointed out, I'm a physician scientist, I'm a cardiologist, and like, Dr. Liu, we have witnessed over recent decades just absolutely phenomenal improvement in terms of the care for patients with cardiovascular disease. So much so, there has been this steady decline in deaths from cardiovascular disease and stroke. And this is witnessed because we have phenomenal, of course, pharmaceuticals. We have interventions. Someone can be born with congenital heart disease, single ventricle heart disease, and we're able to perform complex surgeries. We've got improvements in imaging. There are just really so much that we've been able to bring to bear, and particularly in the cardiovascular arena, we have been able to do clinical trials that provide evidence for medical interventions witnessed on this slide. At the same time, of course, I am reminded by the fact that for other types of diseases, more importantly, neurodegenerative disease and issues around cognitive impairment, they have not had that similar success. And this slide shows sort of the age-dependent effects that will happen in all populations, not necessarily just in the US and Canada, but all populations are going to have this age-dependent increase in cognitive impairment as well as neurodegenerative disease, best typified by Alzheimer's disease. And importantly, what I would also like to point out is that there is actually maybe some bright news on the horizon. This is more recent data taken from the Framingham study suggesting that, for example, the incidence of dementia may actually be decreasing. These investigators tried to attribute this perhaps to particular interventions around, of course, treating modifier risk factors such as diabetes. But you must recognize, of course, in Framingham, there are a limited number of particularly vulnerable groups, including African Americans and Hispanics, where cognitive impairment remains a significant challenge. And we can talk a little bit more, about that. So why did I actually try to tee up this nexus between cardiovascular disease and issues related to cognitive impairment and brain health? And that's because what I really want to do is to communicate to you today that there are some common features. And this cartoon actually helps not only to lay out, if you will, the significant challenges in terms of the financial impact that it has on society, but to talk to you about the science that actually both brain impairment, cognitive impairment, and cardiovascular disease have in common. And we're going to come back more, but this is work that I became intrigued with decades ago. So we all know the central dogma of molecular biology. DNA goes to RNA, goes to protein. And when proteins are made in their unfolded state, they ultimately need to be able to reach their tertiary and quaternary structures. And that's shown, if you will, here on the left-hand side. And that's the role of molecular chaperones. And we'll talk a little bit more about that. But if protein folding is unsuccessful, they can form these amorphous aggregates, oligomers, that can sometimes become amyloid fibrils, and that helps to drive disease pathogenesis. And you're more accustomed to be thinking about amyloid as a consequence of Alzheimer's disease, but I will suggest to you that it has implications for cardiovascular disease. Yet the underlying mechanisms, pathophysiology, and therapies remain elusive in this post-genomic area, or in the era of omics. And so what I am going to hopefully do is to provide and tell you a story about how I became interested in this area. This cartoon, this slide helps to actually convey three essential messages. One, of course, is that after my clinical training, I became interested in science. And science from the standpoint of being an investigator. I had not, for example, Drew, in medical school, done an MD, PhD, which of course gives you a certain kind of training. I, if you will, was a late bloomer. So I came to science a little bit later, but importantly and luckily for me, I was really able to be in the right environment with the right mentor. And I think it's this combination of your environment, your mentor, and your own commitment that helps to drive career development and career advancement. And this little cartoon here is just of my first paper. As you can see, it was communicated by Dr. Lefkowitz, who of course studied G-protein coupled receptors, and along with Brian Kabilka, one of his students, actually won the Nobel Prize. And in fact, Sandy Williams was also a trainee of Dr. Lefkowitz, and we'll get back to you about the story related to that. But the point here is that my interest was fueled by having, if you will, this interaction and this intersection of having a terrific mentor. Now, what I'll also hope for the trainees is that 3 decades later, Sandy Williams is now a friend, he is a mentor, but more importantly, we have been able to sustain that relationship over these many years. And I think that that's really one of the great added pluses of science, in that the relationships that we form are very well-meaning. And of course, I will hope the same for your my trainees who maybe right now might feel a certain way about the folks around you, but do recognize that these could be lifelong relationships and very rich relationships too. So the general concept that I want to talk about is about proteostasis, which of course refers to cellular process that refers to quality control and homeostasis of the proteome. And most importantly, this cartoon just helps to, if you will, provide a little bit more context. Of course, that mRNA, of course, get translated on ribosomes, and there are specific fates that proteins can have. One, of course, could be productive folding. That's actually shown here through the endoplasmic reticulum. Proteins can get packaged and, of course, can be extracellular. But if, of course, the fate might be resulting in unfolded, there are ways for, of course, of dispatching through the ubiquitin proteasome system. And that's actually shown a little bit more in context on this cartoon, where there are key roles for molecular chaperones, be it, of course, being able to package proteins. They can, of course, extrude the cell in exosomes. I described a little bit about the ubiquitin proteasome system here. But as well, there are many chaperones that facilitate, if you will, chaperone-mediated autophagy, as well, of course, there are times when this protein becomes unsuccessful. So the main point here is that quality control of the proteome is the role of the chaperone. And in the era of omics, this, of course, becomes so very much important. And I think that it's so critical for many pathways that, of course, drive disease pathogenesis. So this cartoon actually now tees up one of the main take-home messages that I would like to you. And on your left-hand side is actually a cardiac myocyte. And you know it's a cardiac myocyte because it's stained for sarcomeric actin. This yellow actually is right between, of course, that surrounds this nucleus is actually protein aggregation within a cardiac myocyte. And this is from earlier work taken from Jeff Robbins, formerly at the University of Cincinnati and Children's in Cincinnati. And on the right-hand side, you can, of course, see that these are neurons. You can see, of course, the long dendritic processes, but this is now an extracellular amorphous aggregate or an amyloid fibrils. But what do these cells have in common? Well, it actually turns out that they're post-mitotic cells. And so cardiac myocytes and neurons, because they're post-mitotic and terminally differentiated, therefore will have very low rates of turnover. They're not able to get rid of their protein aggregation issues. So that therefore poses an existential threat for the cell. So therefore survival would be compromised. And so understanding proteostatic mechanisms for two vital organs such as the brain and the heart becomes central to really being able to find relief for these proteins. And we know that there are not existing therapies for these types of proteins. We'll come back to that. Very recently in relation, let me just go back there. There's a drug called tafamidis, Dr. Peter Liu knows this, for amyloid, that basically is a stabilizer for the transthyretin. But other than that, there are very, very few examples for protein for misfolding abnormalities. So we became very much interested. So we were studying chaperones earlier as stress response proteins, but we were quite intrigued when there was this early report, and it was of a sort of French pedigree, which were shown to have, if you will, multisystemic diseases, including cataracts, skeletal muscle, and cardiac muscle. And they basically were just shown to have, of course, so even cataract formation is a protein misfolding problem. And even these aggregates shown here in skeletal muscle, but more importantly, this group was shown through the advances of genetic engineering to be a missense mutation in this particular protein called alpha-B-crystalline. So there was an arginine to a glycine mutation that was able to be identified as the causative event for this protein aggregation myopathy that, of course, causes multisystemic disease. And so we were quite intrigued. So let me just show you a little bit more about alpha-beta-crystalline. This is a cartoon, of course, of a sarcomere. You can obviously see that there are many myofibrils, but the aficionados will easily recognize that whether we're talking about the dystroglycans, the lamin, each of these proteins, they're missense mutations, as well as, of course, causing autosomal dominant disease. All of these, of course, can be associated with various forms of cardiomyopathy. This particular one, alpha-beta-crystalline, actually binds to intermediate filament proteins such as desmin right here within the Z line. It actually turns out that this was a fascinating protein. It actually exists to be close to 3% of the soluble heart homogenate, but when they did knockout studies, a protein that was 3% of the soluble heart homogenate was turned out to be not essential. And that's because there are other members of the small molecular weight family that was able to basically usurp some of its particular functions. Not all, but some. But importantly, overexpression of alpha-beta-crystalline is actually being shown to actually be cardioprotective. So there's a missense mutation, and just to show you just a little bit more in this context, that could be associated with dominant as well as recessive mutations, as well as loss of function mutations. Some of these, of course, are associated with variable penetrance and exivity, but importantly, they have a certain tissue specificity. And so because, of course, this was now a chaperone, it's a heat shock protein, it causes cardiomyopathy, we wanted to basically better understand the mechanisms that would, of course, result in disease pathogenesis. So we approached this question by, of course, performing disease modeling. And what we did under the control of the alpha-myosin heavy chain promoter was, of course, to have, if you will, tissue-specific expression of this mutant protein directly in the heart. This is not an obese mouse, but the clinical types in the audience will recognize that this is a grossly edematous mouse. The aficionados will also recognize that there is significant hypertrophy in the heart that's overexpressing this human alpha-beta crystalline. And in fact, there are these biatrial thrombi. But more importantly, what we were able to do was to identify the mechanistic basis for this particular condition. So we were very much accustomed to this concept of oxidative stress. And for the most part, if you will, there are specific, of course, pathways, biochemical pathways that are driven here by glucose 6-phosphate dehydrogenase that drives the generation of NADPH, such that you have oxidized glutathione that's converted to reduced glutathione by glutathione reductase. So this is a very effective way, if you will, to mitigate oxidative stress. What we were able to identify in this Cell paper was this concept of reductive stress, where what was happening now is that there were excess reducing equivalents in the form of NADPH and GSH that was able to drive reductive stress. And of course, this was the first time that this was identified, linked to a human disease. This is already published work, but we were able to actually show that if we were able to take this, of course, transgenic model that, of course, showing disease pathogenesis, shown here in this green line, and cross it with an animal that actually has G6PD deficiency, we were able to significantly rescue the phenotype. So again, all we did was to basically impact the metabolic aspects related to G6PD that we think was a driving force for reductive stress, and was able to basically rescue that phenotype. So that's a very, I thought, important contribution to the literature. There are a number of studies. The first author for this work was Raj Razazikaran, who is now an associate professor in the Department of Pathology, University of Alabama. We were able basically to show that mutant proteins through a NRF2 KEAP1 dependent pathway is able to drive a number of downstream consequences, resulting in increased levels of reducing equivalence that was able to be both pathologic in terms of protein misfolding, as well as for hypertrophic cardiomyopathy. And I think that this work has been reproduced by several other laboratories. Just as importantly, we, of course, took advantage of other types of disease modeling. I'm showing you here for human iPS cells. Of course, this seminal work that resulted in a Nobel Prize for Shinya Yamanaka, in that if you were able to just take somatic cells, apply, if you will, these factors to those somatic cells, you're able now to generate human iPS cells that under particular conditions can be coaxed to become a hepatocyte, a pancreatic cell, a cardiac myocyte, or a neuron. So we've actually, of course, used human iPS cells to see if we can better see whether, of course, this human iPS works. So again, using fibroblasts as a somatic cell as a source, we were able to then model those fibroblasts to show that, of course, this work could be significantly recapitulated. So not only did we recapitulate protein aggregates, but we were also able to recapitulate the hypertrophic cardiomyopathy. And this is actually shown here by Pat Limfeng, who was a postdoctoral trainee in the lab. And we also, of course, then went on to do a variety of additional studies with other members of the protein chaperone family. This is Katie Mitzenfeld, who's now an assistant professor in the department at Washington State University, and several other members in my lab. This, of course, is Shuping Lai, who is a research scientist in the laboratory. And Michael Rydell is actually now in industry. So the point I'm making here is that we were able to do a variety of studies doing disease modeling that has resulted in further enhancing our understanding of these protein folding myopathies. And no talk is complete without showing you, of course, the fact that you can, of course, model these agents and be able to generate beating cardiac myocytes in the setting. So that's, if you will, the first part of my talk. Dr. Mulvihill shared with you earlier that, of course, I'm not able to be with you in person. This invitation came along well over a year and a half ago. And that's because, you know, many of us are still recovering from this pandemic. And of course, since the introduction of SARS-CoV-2, which of course is a respiratory virus that causes, of course, a number of significant consequences, not the least of which is that, of course, you've got viral entry into the lungs, but just as importantly, that viral uptake then results in a variety of downstream consequences, activation of the immune system, many other pathways. There are then many downstream consequences. There are even reports that as much as 40% of patients may have cardiovascular dysfunction. There's acute renal injury. And just as well, this is, of course, not only disrupted our lives, be it our research, and I just want to basically shout out to the trainees that you just got to hang in there. We're going to get through this. It clearly has disrupted your plans, many, many plans. But, you know, there's also perhaps, you know, an upside. And so what I wanted to just do is to just have a lighthearted moment here. So one of the things I like to do with my trainees is actually take them to lunch. And so since you're obviously not able to take your mask off, I decided to, when I come on service, to have a little competition. And the competition had to do with, you had to wear, you know, your crazy socks and tell a story about the socks. And it actually turns out that the medical students absolutely love this because, you know, everyone could have wear their socks. As you can see, I like to wear my socks too. I'm not obviously in the competition, but the medical students on the service just, they killed it. And so many of these pictures here, you can see they got a gift card. That was my gift to them, of course, but I didn't have to pay for lunch, but they were still there. So we were actually able to, you know, as they say, when you have lemon, you got to make lemonade. And so this is just a moment to just share with you that we actually been able to survive a pandemic, and you will be able to do so. All right, so back to the story around, of course, COVID. So we became interested in COVID, and what I want to do is to basically shift the emphasis from the idea that, of course, one of the ways that SARS-CoV-2 gains entry is through the angiotensin II converting enzyme receptor, to also make the point that there is also another member of the heat shock protein, GRP78, that also is associated with, if you will, viral uptake. And this is actually shown more in this cartoon. So here you have this left side, this divider down the middle of the cell. You're familiar with the fact that SARS-CoV-2 is uptake by ACE inhibitor, but what I want to also introduce to you is this protein that's called GRP78. So GRP78 is a member, it's a 78 kilodalton protein. It resides in the endoplasmic reticulum. It plays a key role in sensing ER stress. And that ER stress then results in activation of the unfolded protein response to result in upregulation of a series of molecular chaperones to basically deal with ER stress. In a complementary way, there is also, if you will, the heat shock response that's classically driven by members such as HSP70, and you'll hear a little bit more. But here, these are regulatory pathways. They bind to upstream recognition sequence called heat shock elements that results in upregulation of heat shock proteins. So these are complementary pathways that respond to protein misfolding. They're able to, of course, cope with, of course, cellular stress that might be, of course, impacting a cell. But just as importantly, upregulation of heat shock proteins will be shown to be able to affect a number of pathways, including pro-inflammatory cytokines, prevent apoptosis, as well as, of course, mitigating issues such as fibrosis. So this is a key and important way in which cells are able to respond to cell, not the least of which I should point out to you that co-receptor translation of even SARS-CoV-2 actually takes place on the endoplasmic reticulum. So this is a key and important molecule. So we were very much interested in better understanding what might be the consequences of a particular chaperone, again, this ER chaperone. And what we actually did was to have a collaboration with our pathologists who actually were able to identify in patients who are dying of SARS-CoV-2, massive upregulation of GRP78 in the lungs. And you can see that this is shown here in brown, the SARS-CoV-2 protein, but probably a little bit more pictorially is shown here in panel A, where we actually looked in pneumocytes. So you can identify pneumocytes, not shown on the slide, with a pan-keratin stain. And it's quite interesting when we compared neurocytes here in panel A to macrophages here in panel B, that there was this distinct difference between SARS-CoV-2 driven upregulation of this ER chaperone compared with controls. And controls were basically individuals who may have had lung resection compared to a little bit more of the overlap, for example, that you would then see in macrophages. So we think that we are onto something here in terms of the tissue tropism that these particular, and it kind of makes sense. You know, you've got massive upregulation of the ER stress response, and it's very clear that there are particular cell types within the lung. And this, of course, has implications for other cell types, of course, in the heart, and we're going to be, of course, doing those types of studies. In parallel, we became quite intrigued to see whether or not we can begin to start measuring. So the same way that you know that cytokines are increased in COVID, obviously we know that there's dysregulation of a number of immune pathways, IL-6, many others have been measured. Of course, CRP, many D-dimers. We also had a chance, if you will, to look at plasma levels of these various heat shock proteins. And this is quite interesting. So we were able to follow a cohorts of patients who were getting convalescent plasma, but importantly, they were able to actually get their plasma drawn at day 4, 7, 14, and 28 days after, if you will, getting convalescent plasma. And so what we're just doing now is just measuring for HSP70, and you can see that there is sort of this, if you will, temporal decline. So if you see, for example, patients who have survived ARDS, they're actually shown here, but then there are also patients with ARDS who have died. But importantly, if you recover, there is this temporal decline in HSP70. And that kind of makes sense. So this is the classical heat shock response. On the other hand, we also looked at this GRP78. As I mentioned to you, there is this ER chaperone. And I just wanted to sort of just point out, so here is a patient with ARDS. We were able to basically find from the clinical record that this was a patient who was doing well, as you know, in the first 7 to 11 days. There are patients who were reported to be improving with COVID, but then they had this precipitous decline. And in the hospital, we were pretty much able to track, if you will, what was happening with GRP78 that matched this particular patient who was recovering, had a relapse, but then subsequently declined. So we think that really being able to follow plasma levels, we can talk a little bit about the source of these HSP70, as well as the other members of the GRP family, might actually provide new insights in terms of being able to provide. And so this is just another way of us looking at that same kind of question where we've now plotted HSP70 to GRP78, and you can clearly see that there were subsets of patients who, who have died, who had these high levels. And we think that this is being driven by low levels of GRP78. In other words, there could be dysregulation of the unfolded protein response compared to patients who had no ARDS or no COVID. And I should point out to you that some of the controls include myself, because of course, we just didn't have access to as many normal volunteers. But the point here is that we think we're beginning to get a signature, and there's a lot more that we can begin to learn about these, if you will, proteostatic markers in the context of COVID, which again makes the point to the students that as much as you've got to basically focus and you've got your primary projects and you got to get that done, there's really going to be quite a bit that we still have to learn about an agent that not only impacts the lung, but of course has multi-systemic effects. And so these are just some of the questions that we're interested. What might be driving the tissue tropism, as well as the plasma HSP biomarkers, what might be the consequence of COVID and even sex on GRP, HSP70 and GRP biomarkers. And last but not least, we think that there's also a consequence having to do with aging. So this was not work 15 months ago when you first sent me my invitation, but that's the cool thing about science. You're actually able to pivot, and we've got some robust experiments that are ongoing. So in the final part of my talk, I wanted to basically tell you a little bit more about my passion. Dr. Mulvihill shared with you that, one of the things that I've really found to be incredibly exciting, and I know that Dr. Liu will agree with me, is that in addition to being part of a community, science is just absolutely amazing. I've been able to visit all parts of the world. And obviously, you don't sort of run for office to be president of the American Heart Association. This is an honor that your peers bestow on you. And so through those activities, I became particularly passionate about, if you will, health disparities, just really finding that there are clearly populations where our collective efforts, so whether you're a basic scientist, you're actually helping to address understanding the pathogenesis of disease that will ultimately be able for us to really meet a future state of health equity. And just as well, I think that we are taking much more bolder steps to better understanding how we can basically improve healthy life expectancy. So it's more than just, if you will, life expectancy, but you want to have healthy life expectancy. What that means that you're basically trying to do the best that you can to reduce morbidity. You want to be able to live a long life, but free of morbidity. And just as well, these are some of the factors that are helping to drive health equity, as well as, of course, the social determinants of health. So at the Cardiovascular Center here at the Medical College of Wisconsin, we have organized ourselves about what we call signature programs. They could include ATVB for arteriosclerosis, thrombosis, vascular biology. We have cell biology and heart failure, hypertension, and precision medicine. Cardio-oncology was our most recent addition, and I don't know if the announcement has been made, but they have just successfully been able to get an AHA, strategically focused research network. But importantly, what I'm most proud of is how we've been able to think what I like to call going for the goal. And so we organize ourselves as affinity groups. We then are able to, if you will, apply. And of course, we're in the summer just before the Olympics. So we have a way of, you can be in a bronze signature program, a silver signature program, or a gold. And so if you're bronze, it's an affinity group where you've got your R01 grants. A silver program is where you now have PPG grants, these larger grants, such as strategically focused networks. But more importantly, if you're able to basically touch on what we call this T0 to T4 continuum, T0 being basic research, T4, T5 continuum, where your particular intervention actually goes and impact communities. That's what it means to be going for the goal. And so in our program, it's still aspirational, but the point here is that this is probably what is the most exciting part of science. And the foundation of that, in my opinion, is basic research. But that basic research must, if you will, have a clinical component, a translational component, and it ultimately needs to be able to impact communities. So we're very proud of our T32 training program. This is an NHLBI program. We work very vigorously at putting trainees at the center. While it is true you may have one mentor, my advice to you is to have multiple mentors. And none of your mentors should ever, if you will, leave your Rolodex. Just accrue as many mentors as you can. And the point here is that we try to make trainees the center, and we take a multidisciplinary approach to basically driving mentoring, as well as, of course, seeing that our trainees, if you will, take fullest advantage. So this is a T32 program that's also supported by philanthropy. We continue, of course, to see this as the foundations for our future success. The other point I want to basically be able to share with trainees is that you're going to be living in a data-rich environment. So the more you're able to either yourself or with others be able to work with large data sets, it will just make it so much more impactful. So whether you're basic in clinical research, having trainees, and this is one of, I think, the masterstroke of the American Heart Association, is that trainees can not only, you may have a basic project, but you have to become familiar with a translation project. You have to become familiar with a clinical project, because I think that you'll be much more impactful, ultimately being able to bring and enhance not only the care, but the outcomes for our patients. And just as well, I think that part of what we really want to be able to do is to just bring as many into the fold as possible. And this is part of my, if you will, my AHA presidential talk, which has to do with the future of diversity. So as part of my work with the American Heart Association was just begin to ask ourselves, well, where can we be able to have the most impactful efforts to increase the number of underrepresented members from racial women and racial and ethnic groups into science? And we decided to basically focus on the graduate years. And the reason for that is shown actually in this slide, and that if there are actually students, be them white, Black, or Latino, obviously I'm talking about in the context of the US now, but when they are actually STEM graduates from high school, they show up and declare that they would like to be STEM majors at the same rate as their white counterparts. But when you then look at graduation rates over the course of 4 years, you see there is a lot of leakage in the pipeline. And so the American Heart Association is investing, and we are one of the centers in the summer enrichment research. So I'm sure that at your institution, you have an infusion of trainees who are coming into your ranks, because of course they are between their academic years. But that's an important contribution that you can be part of. And I will encourage you to certainly do that. I've been extraordinarily fortunate. I had a chance to be invited by the Journal of Clinical Investigation to be able to tell my life story. And what Christina is going to do is to throw into the chat a recent perspective article that was just published in the Journal of Clinical Investigation. But, the story talks about this, sort of line that, science is a discipline with no borders. And I feel there's, as much as you're north of the border, I feel that my journey in science is one where it's been incredibly enriched by people from all walks of life. And I will certainly, and so you'll get a chance to read, it's a quick read, but this is when, of course, you could see that, my hairdo looks a lot different to now, but that's okay. It's really been a fantastic ride. And this is actually, one of the real heroes in my life. This is Dr. James Morrell, my great uncle, who, so I'm not the first physician in my family, but he got his medical degree from the University of Edinburgh in 1921. Yes, you heard it 100 years ago. So they are heroes in all of our lives, and we need to really be able to celebrate them. I shared with you a little bit more about mentors. I was blessed and fortunate to be part of this AMOS Medical Faculty Development Program as a fellow, and it certainly has been able to contribute. But just as importantly, when I matriculated into Johns Hopkins in 1978, there were only about 1,400 African-American men applying to medical school. Fast forward to 2014, that number had actually slightly declined. And that's a challenge for all of us. And so I'm absolutely committed to seeing how we can increase opportunities, because I think that diverse teams do extremely well. And this is not just opinion, but there is evidence in the literature to sort of support that. And a key and important part, of course, is mentorship, for which I know with colleagues and friends right there at University of Ottawa, you have superb mentors. So as I sort of bring this talk to a close, I would like to suggest to you that being able to ensure that science is able to embrace and have voices from all backgrounds will all make us be stronger. This advocacy, I think, is critically important, particularly with respect to STEM and implementation research, because I think that these are some of the key drivers for really being able to achieve health equity. And just as well, I think that this inclusive excellence is critically important for us to really be able to transform cardiovascular research, both at the basic, clinical, and of course at the population levels. I would be remiss if I did not try to tell you a little bit more about some of my heroes, but I'm sure you recognize that this is Wayne Gretzky, and one of Wayne Gretzky's famous quotes is that you will miss 100% of the shots you never take. But figuring that you've already heard that, let me leave you with a few more pearls. These are sort of Ivor Benjamin tips of the day. Rule number one, never disregard your highly reproducible or unexpected results, okay? So take that as a tip. I've got two more for you. Rule number two, provide sufficient compelling evidence to convince your third reviewer. You know who the third reviewer are. Yes, of course. And last but not least, science is a lot more fun when you're challenging existing dogma. These are sort of some of the rules that I try to, or at least I try to live by. I'm sharing them with you, and you're more than welcome to share them as you please. So again, Dr. Mulvihill, before I turn it back over to you, I just want to express my thanks and appreciation for the many colleagues, trainees, patients, as well as, of course, this remarkable community in cardiovascular science research. And I'm so, of course, grateful to our many funders. And last but not least, thanks to you again for the invitation. Thank you so much.
Well, thank you, Dr. Benjamin. That was a really terrific presentation, covered a whole wealth of new data and also questions of diversity that we can certainly apply, as well as how to support our trainees. I think it's just terrific. And I'll also welcome Sabrina Robichaux, who is a Vanier Scholar in the PhD program at the University of Ottawa. So she is the chair of our trainee committee, and she's going to help with the question and answer. Okay, so
thank you so much again, Dr. Benjamin. That was a fantastic talk.
You're welcome.
We'll start with Dr. Liu, who has a question. Dr. Liu, you can just unmute yourself.
Yeah, so, oh, let me just see, yes. So that was just fantastic, you know, so it's so inspirational, and blending the science and the mentorship and, you know, sort of the translational aspect is just so outstanding. It's unbelievable. And so just as you know, and so I think you'll be sorry that maybe you came, is the fact that we are in the process of applying to CIHR training grant. And so we are going to ask you as, if possible, down the road as an advisor, you know, for our program. I think it will be so exciting, so, you know, so fantastic, you know, because you have both the content expertise, the diversity, you know, sort of aspect, which is so important for our application. And but at the same time, you know, the mentorship experience is just so valuable. I do have one kind of a question, that is that, you know, I love the proteostasis concept. Of course, you know, we are a huge fan of that, and also your work on the of the crystalline data. And so, you know, the question is, you know, how to potentially use that mechanism as a way to address, I think, you know, certainly things like HFpEF, you know, sort of cognitive impairment, all these processes, you know, I absolutely agree with you. And so the so I think you demonstrate nicely, certainly redox manipulation, probably is playing a role.
Yep.
But I seem to remember earlier that one of the publications was the fact that the exercise, can also benefit. And of course that plays into nicely, in your prevention, the whole journey of cardiovascular disease as well. And I quite often use that story, to encourage, our teams, but also patients, that exercise so important, can potentially counteract aging. But I could never explain, sort of exactly what exercise does, in the setting when, it's such a kind of a challenging condition, right? Nobody ever says that. I know we now have this controversial drug, for Alzheimer's from FDA, but nobody's really convinced. But the exercise certainly does. So so I'm so curious as to what you think that the exercise, might be helpful in, sort of, in the proteostasis space.
Well, first of all, that was very kind and very generous of you. And, since you have been, and, Sabrina and others have been such gracious hosts, how can I say no to an invitation on Zoom, right? So that was good. All right. So your question about exercise, I think helps us to think more broadly about, things that we know and things that we don't know, right? I exercised this morning, okay? And at the same time, so what were the benefits that I got? Well, for sure, there is good evidence just purely based on just managing a common risk factor such as hypertension. For people who are pre-hypertensive, there are good data that said if you were to basically put 100 people on an exercise regimen, a third of them do not go on needing medications, right? So there is definitely some benefit that you can get just from exercise. But I think that particularly in the area of brain health, we know about exercise on vascular function and everything else, but I think that there are data to suggest beyond, if you will, ApoE4 being a risk factor for Alzheimer's disease, or probably the most important genetic risk factor, while ApoE might be neuroprotective. We certainly know that people who have more exercise in their regimen lifelong seem to have less in the way. So these might be correlations. They're not necessarily causal. So I don't, it's a life's experiment. So I didn't figure that, I will live my life and then look back and say whether or not I should have exercised. So I do it anyway. The final point I want to make is that it was Jeff Robbins. I want to give credit where it's due. So Jeff Robbins did take that same alpha beta crystalline mutant mouse, put those mice on an exercise regimen, and they were actually able to show a protective effect. And so the way that I would think about that is that the exercise was basically able to counterbalance, if you will, the dysregulation of the pathways.
All right, we have another question from Dr. Katie Rayner, who says, what a terrific presentation, Dr. Benjamin. I am wondering if HSPs contribute to some of the sex differences we see in cardiovascular disease or neurodegenerative diseases. HSP have so many hormone responsive function, wondering if they are linked.
That's a terrific, that's a terrific question. And I'm going to give you two responses. Well, I'm going to give you a response in two parts. The first part is published data, and the second part is a little bit of a hint of some things that we're thinking about. So it actually turns out that HSP27. So this is a 27 kilodalton protein. You can write it down as like, so because there's a whole family of small molecular weight heat shock protein, this would like be HSPB1. It actually turns out that HSP27 contains an upstream estrogen response element. And so there is the possibility that hormonal balances may actually regulate HSP27. So again, more association than causal, but that's definitely one. And in fact, a colleague of mine, Dr. Ed O'Brien, who used to be at the University of Calgary, has really been one of the leading authorities in this area. In work that we haven't published yet, we are actually, now that we're able to look at these plasma markers, we're actually doing, and we are looking preliminarily at the influence of, sex. And so we were basically doing this more in preclinical models, in animal models, but this also has to do with some of the ApoE genotypes. So stay tuned. Very insightful question. And that's why we do research, right? So great point.
All right, thank you. With that, if anyone has a last question, now would be the time to send it either via the Q&A or with the raise hand function at the bottom of your screen. Dr. Liu.
Yeah, so if I can be spoiled with a second question, and I was really fascinated by the GRP78 data.
Yeah.
And that is so exciting, because it really shows the involvement of the stress response system. Yeah, exactly. The protein unfolding component, right, as critical both for the viral infection and also, of course, the dysfunction that we may actually see in that type of setting. So I guess the follow-up questions are that one is that does the virus actually take advantage of those components, as part of its biology, as a as a component for both its survival and of course altering the environment for its own benefit. And then the other aspect was really interesting that you actually see that in the bloodstream, as a biomarker. And so the question is, are these you think these are proactively released, as a kind of a reflection of the cell stress, so that they are, maybe packaged and, sort of sent out, or do these represent, cell damage itself? Because I was thinking that it may be actually a signaling component. Yeah, just some thought on this intriguing data.
Yeah, very, very, very insightful question. Actually, your first question gives me the opportunity to share some information that I didn't during the seminar, which is that viruses themselves don't have heat shock proteins. So what a virus must do is to come into the cell and co-opt the protein folding machinery of the cell. And so viruses not only have proteins that might turn off certain types of pathways of the cell's machinery, but you're right. The reason why it goes on to be lytic is because the virus has co-opted the cell's protein synthesis machinery for its own benefit. So cells themselves have to fight back. So how cells have that delicate balance, to me, is life and death. Okay. So now the other part of your question in terms of, because before people will just look at heat shock proteins, they will take the cells, they'll grind it up or whatever. But we are now beginning to see that maybe these cells might actually have a signature in plasma. And they're going to be both a combination of dead cells. We think that that's going to get released. We also think they're released in exosomes. I kind of give you a hint about that, right? But just as well, there may be damaged but not dead cells the same way. So the reason why I think these HSP biomarkers are so amazing, because as you know, as a cardiologist, we measure high sensitivity troponins. But that's just released from a cell. It doesn't give you any insight about the molecular pathway that is dedicated to repair after injury. And that's what proteostasis markers are. So on the one hand, it gives you a signal, oh my goodness, it's stressed, it's injured, but it also might give you an insight about your repair mechanisms and the efficiency of those repair mechanisms. That's why we're very excited about it.
All right, and I think with that, I think we're gonna wrap this up right now. Big thank you to Dr. Benjamin, who agreed to join us today for her seminar. That was a wonderful presentation again. I think we definitely picked the right speaker to end this year's weekly research round.
well, you're very kind. And once again, to each and every one of you, to you, Sabrina, to you, Aaron, to you, Peter, you've been fantastic hosts. And for all of the other trainees who were able to join us today, again, it's a singular honor. Thank you.
I'd also like to remind the trainees that we are going to have a mentoring session right after this on a different Zoom link, so we can join right now.
Sorry, right. Peter, you have to go back, and you're not a trainee anymore.
Thank you again. Take care.
See you at sessions. Bye-bye.
Thank you.
Thanks.